Alright, this is my second video essay for the Module 2 unit, which is muscle and sensation. So let's start with the chemoperception of our food, which is called gustation. So gustation takes place in lingual papillae, which are found on the tongue. We have a few different types of lingual papillae. We have fungiform, filiform, foliate, and circumvallate. And these lingual papillae contain, inside of them, they're going to contain taste buds and then the taste receptor cells. And then at the basal level of these lingual papillae, we're going to have a nerve synapse there. So there's 5 different types of taste that we can perceive. We've got sour and salty, sweet, bitter, and umami. And there's like 2 different types of gustation pathways based on these taste perceptions. So starting with sour and salty, if we eat a food that contains a salt or a hydrogen ion, which is acidic, we're going to take this sour and salty pathway. And this is the most efficient. So right here on the membrane of this taste bud, we have what's called an ENaC channel, which allows these ions to pass through, either a hydrogen ion or a sodium ion. And that's going to cause a depolarization of our cell, which is going to activate this voltage-gated calcium channel, which allows calcium to come into the cell, which is going to cause exocytosis onto our neuron, and that is going to be sent to the brain as a taste sensation. Another way that we can release calcium into our cell is by, there are these potassium leak channels that are on the membrane of this cell, and this hydrogen ion fits nicely in there. So it can go in and plug this potassium leak channel, which will cause depolarization of our cell, which our voltage-gated calcium channel will be sensitive to, which will lead calcium coming into the cell, and that will cause our exocytosis of neurotransmitter onto our neuron going to the brain. And then our next taste perceptions, we have sweet, bitter, and umami. And these use a G protein couple receptor. So once again, on those taste receptor cilia of our taste bud, so we have a G protein coupled receptor on the membrane of that. And what's going to happen is a sweet, bitter, or umami taste particle is going to bind as a ligand to that G protein coupled receptor. And the it's going to be the alpha subunit that dissociates from this G protein coupled receptor, which is going to activate phosphodiesterase. And this cleaves a phospholipid off of the membrane, turns it into DAG and IP3. IP3 is used as a second messenger to basically do one or both of 2 things. It can go ahead and activate a calcium channel, a ligand-gated calcium channel, to allow extracellular calcium to come into the cell, or we can send IP3 down to the smooth endoplasmic reticulum, which is going to release calcium from the SCR. So, and we can also do both these things. We can send IP3 to the smooth endoplasmic reticulum or a membrane channel. All that matters is that we get calcium into the cell, which is going to cause the release of neurotransmitter and send that signal to the brain. So the innervation here that we're dealing with, so the nerves that perceive taste are going to be our facial nerve, our glossopharyngeal nerve, and our vagus nerve. So these are going to be the nerves that send these taste sensations to our brain. And different taste sensations are distinguished by the proportion of action potentials from different receptor types. So if we have 5 receptors and we have like 3 sour tastes we have like 3 sour taste receptors that are stimulated and then like 2 umami, we're probably going to perceive more sour there because we have a higher proportion of action potentials coming from that sour receptor type. We can also distinguish different taste sensations with the help of olfactory input, which is our smell. And then we also get additional sensory input, like from the texture of the food that we're eating. So we just wrapped up gustation. Let's go ahead and move on to olfaction here. So say we have an odorant. So this would be like a gas particle that's mixed in with the air. We breathe it in through our nose. And then this is a little bit of the anatomy here of what's going on up there in our nose. So we have this mucus layer that is secreted by this olfactory epithelium. So those epithelial cells are secreting this mucus, and this odorant is going to go in and it's going to dissolve into this mucus. And in this mucus lie dendrites of olfactory nerves. They're these sensory nerves that are going to pick up on this odorant, and then through a series of events, we're going to end up sending an action potential to the brain. So I blew up one of these olfactory neurons, and the dendrites of these neurons are lying here in this mucus layer secreted by the epithelium. And then, so here, what we have happen is, say our odor particle is going to bind to a G protein coupled receptor as a ligand, and then that G protein coupled receptor is going to undergo a conformational shift, which is going to activate adenylyl cyclase. So now we're going to produce cyclic AMP, which is going to serve as a secondary messenger that is going to open up a calcium channel. And then that calcium coming in stimulates a voltage-gated chloride channel, which is going to further depolarize our cell because this chloride is going to leave. And this calcium is coming in, so we're depolarizing. And then that's going to stimulate the release of neurotransmitter onto our olfactory bulb. So those dendrites come down into the mucus layer, they travel up through the epithelial layer, and then they travel up through a bone towards the top of our nose, right there at our skull. And then we're going to synapse onto the olfactory bulb, and we're going to send that olfactory sensation to our brain based on that odor particle there. Okay, so that wraps up olfaction and gustations. Now let's go onto our auditory sensations. So how do we transmit sound waves into electrical action potentials? Well, it's very complicated, as you can see, so I'm going to do my best to try and explain this. So starting with the nature of sound. Sound is basically compressed air. It's like alternating wavelengths of compressed air and decompressed air. And the wavelength is determined by how many peaks of amplitude you have in 1 second that pass a certain point. So if we have a lot of these amplitudes, we have a high frequency wavelength, which is measured in hertz, which will be perceived as a high-pitched sound. And if we don't have a lot of peaks passing that point in 1 second, we're going to have a low-frequency sound, which is going to be perceived as a low pitch. And also the loudness of the sound, how that's perceived, is based on the amplitude of our sound waves of compressed air. So if we have these big amplitudes, we're going to have a big loud sound. And if we have little amplitudes, we're going to have a quiet little sound. So these sound waves get channeled into the ear by the external auditory canal. And if you look at your ear, right, you've got that interesting shape. So that's going to help to funnel these sound waves into your auditory canal. And the external ear is separated from the middle ear by the tympanic membrane. And the tympanic membrane is going to vibrate when it receives these sound waves. And then we cross over into the middle ear, which has, the defining feature of the middle ear are our ossicles. We have the hammer, the anvil, and the stirrups, basically. So what's going to happen is the tympanic membrane is going to vibrate these three bones. These are actually bones. It's going to vibrate them. And then in turn, basically the way this works is we have at the end of the stirrups, we have what's called the oval window, which begins our inner ear structures. So it's the oval window of the cochlea, which is the inner ear structure responsible for our hearing. That's where we have our action potentials being produced. But what's interesting here is that the tympanic membrane has a surface area that is 20 times greater than the oval window. So from the tympanic membrane vibrating those ossicles down into the oval window, it's going to serve as, it's really going to concentrate that energy onto the oval window and amplify that signal. So now in the inner ear, we have the cochlea, which lies in the temporal bone. And basically the cochlea consists of a few structures here. We have an oval window, a round window, and then we have this spiral structure that is comprised of 3 chambers. We have the scala vestibuli, the scala tympani, and the middle chamber, the scala media. And so running up all the way through this cochlea, through these channels, we have what's called the basilar membrane. And if you look at this little diagram that I've drawn here, the basilar membrane has sections that are more innately sensitive to low and high frequency sounds. I actually should have switched these around, the low and high frequency. And they progress as you go up the cochlea. And so this produces a phenomenon called resonance, which is when frequency of sound waves matching up with the same innate frequency of the basilar membrane causes the basilar membrane to vibrate, which will be important later. So if we take a cross-sectional view of this cochlea here, we see that we have the scala media, scala vestibuli, and the scala tympani. And then the 2 chambers on top of the scala media. So there's the scala vestibuli and the scala tympani. These are full of a fluid called paralymph. And the scala media is full of endolymph. And then in this cross section here, you can see this little organ that I drew. And if we zoom in on that, that's called the organ of Corti. And it consists of numerous different structures. So we have the vestibular membrane that is separating this from the scala vestibuli. We have the basilar membrane, which is separating us from the scala tympani. And then we have the tectorial membrane. And then we have this epithelium with these hair cells in here. And this is where our action potentials are going to be produced. So these hair cells are connected to this tectorial membrane and to the basilar membrane. And there's an outer row and an inner row. And what happens over here is we get these fluid waves that are caused by our stapes, or our stirrups that I mentioned earlier. It's that bone. So when the tympanic membrane vibrates these bones, it causes a fluid wave that goes from the oval window all the way through the cochlea. And then at the other end, there's what we call the round window. And this acts as a pressure release valve. So as the oval window goes in, the round window comes out. And this helps facilitate these waves that go through the cochlea because fluids can't be compressed, right? So we need an elastic structure that has some give to it to allow us to have these waves that can happen. So I've blown up this hair cell here. So we have this fluid wave that's caused by our stapes in our oval and round window. And this wave is going to hit these little hairs that are coming off of this hair cell. And now so these are actually microvilli here. So they have a plasma membrane. And on this membrane, you have these mechanically gated ion channels. And they're connected to each other by this little string that connects all these microvilli. So when a fluid wave hits this longest hair, and they come in an ascending or descending order. So one is taller than the next one's taller than the one before. And then basically when these waves come, they bend these hairs, and since they're all connected, that causes these mechanically gated ion channels to open up, which is going to allow potassium to flow into the cell, which is going to depolarize our cell, which opens up our voltage-gated calcium channels. And this is going to stimulate the release of neurotransmitter onto our cochlear nerve, which is going to go and be processed in our auditory cortex of the brain there. So potassium rushes in because the electrical gradient has a strong influence on potassium in this case. The endolymph that these hair cells are bathed in is full of potassium. And so to explain this, we have our endocochlear potential, which is the result in the difference of charge between these three chambers, and also the cytoplasm of the hair cell and the endolymph. And so this contributes to a high driving force for sodium to enter, or not for sodium, but for potassium to enter into the cell. And this is created by a system called the stria vascularis, where we're basically using a sodium-potassium pump and a potassium chloride and sodium channel. And we just repeat those twice. And so we secrete sodium, or we secrete potassium here into this endolymph and create a high concentration of it, which creates our endocochlear potential, which allows potassium to flow into this cell here and depolarize it. So if we come back here to our organ of Corti, we see we have an outer row and an inner row of hair cells. So our outer rows are going to receive these vibrations, and we have a structure here on these hair cells called Preston, which basically pulls the basilar membrane up and this tectorial membrane down, which is just going to make our signal more sensitive. And that's going to create a wave from this outer row that travels to the inner row. And that wave causes the mechanically gated ion channels to open up on our inner row, which is going to depolarize, right? And it's going to allow calcium into the cell, which is going to cause our neurotransmitter to be secreted onto our synapse. And that's how we turn waves of sound into electrical action potentials. So that's pretty cool. I didn't think I was going to be able to explain that, honestly. I was studying this for hours, so I'm glad that worked out. But anyways, let's move on to the visual system. So first, let's go over some anatomy of the eye here. So we have the posterior chamber and the anterior chamber. The posterior chamber is filled with vitreous humor, which is like an egg white kind of gelatinous substance. We have a retina in our posterior chamber, which contains our photoreceptor cells, bipolar cells, and our ganglion cells, as well as our optic nerve, our fovea, and all these structures that are going to be important for interpreting light. And then in our anterior chamber, we have the structures that are responsible for refracting light into the right spot onto the retina so that it can be interpreted and sent to the brain as action potentials. And the anterior chamber is filled with aqueous humor, which is a watery substance. And we have our two refractive structures are the cornea and then our lens. And in this anterior chamber, we have these suspensory ligaments that hold the lens up. And this is where you also find the pupil and the iris. And the iris and the pupil is just a hole in the middle of the iris and these ciliary muscles that allows light to come in and be refracted by the lens and hit the retina. So here's kind of a frontal view of the lens and ciliary muscle, construct. So you have these suspensory ligaments that hold the lens up, and you've got this ciliary muscle that acts on these ligaments. So when we are looking into the distance, that ciliary muscle is going to relax, which pulls those suspensory ligaments taut, which is actually going to flatten out our lens. Because our lens is kind of it's a malleable structure made of a transparent collagen. And so we can kind of bend and mold it using this ciliary muscle. And then when we're looking at near, our ciliary muscle contracts, which causes our lens to thicken. And then we can refract that light the right way onto our retina. And those suspensory ligaments are going to go slack because we've contracted the ciliary muscle, so those suspensory ligaments aren't pulling on the lens anymore as hard. So we're going to have a widening of that lens. And this phenomenon of our lens changing based on the distance, the thickness of our lens changing based on where we're looking, is called accommodation. And it has to do with the way that light is hitting our eyeball. So an important term here is refraction. And this is where we bend light using the cornea and the lens onto our fovea. So some refractive errors that you can have are what we call myopia, which is where you have a long eyeball, and this is where the light crosses too early onto the retina. So because you have that long eyeball, this light it's gonna cross too early. It's not gonna hit the fovea. It's not gonna be focused on the fovea, so it's gonna focus too early. And myopia is trouble seeing at distance, but we can see okay up close. And then in a normal eye, we're the right eyeball length. We don't have any asymmetry on the cornea or the lens. And so that light is going to refract just right onto the retina. And then in hyperopia, we have a short eyeball, and that light is going to focus too late. So we're going to have trouble seeing up close, but we'll be fine up, far away. Presbyopia is a condition that is caused by our lens losing elasticity, and this causes difficulty accommodating. And then we have astigmatism, which is a refractive error that is caused by asymmetry in the cornea, lens, or both. That's going to affect the refraction of light and make it hard to see. So we've discussed how light gets refracted onto the retina. Now let's talk about phototransduction. So now the question is, how do we turn light into action potentials that our brain interprets as vision? So you have light, and light has these little particles called photons. And on our photoreceptor cell of our retina, we have a pigment called rhodopsin, and this is going to absorb light. And those photons, and within rhodopsin, there's a structure called 11-cis retinol. And when we add a photon to 11-cis retinol, it becomes 11-trans retinol, which is going to cause it's going to cause a conformational shift where this changed form of retinol is going to dissociate from rhodopsin or transducin. I'm not quite sure what the name of this G protein coupled receptor is. I think it's transducin, or maybe transducin has rhodopsin in it, but this is going to cause this G protein to become activated. So this G protein coupled receptor called transducin is activated by light, and it's activated by 11-cis retinol becoming 11-trans retinol and dissociating from the protein. So transducin is going to activate Okay, so let me preface first. So we're talking about conditions of light, right? We have light because things are different in the dark. So when we have light, transducin is going to activate phosphodiesterase, which break down cyclic mononucleotides. And we just happen to have here on the membrane of this photoreceptor cell a sodium channel that is being held open by a cyclic mononucleotide. So if we activate phosphodiesterase, this cGMP, cyclic GMP, is going to be broken down, and that's going to cause our Na channel to close, which is going to cause this voltage-gated calcium channel to close, which is going to stop us from secreting glutamate onto our bipolar cell. And if we stop secreting glutamate, so this bipolar cell has a G protein coupled receptor that receives glutamate at its ligand to tell it to inhibit this calcium leak channel. So if we're not secreting glutamate anymore because of the effects of light on 11-cis retinol, then our calcium channel that we have here is going to be, it's always open unless it's being inhibited by this G protein coupled receptor. So in the light, this calcium channel is open, and that's going to cause the release of glutamate onto this neuron, onto this ganglion cell here, which is going to leave and go to the brain via the optic nerve, which is cranial nerve 2. So that's what happens in the light. But in the dark here, we don't have this G protein coupled receptor activated, which means that phosphodiesterase is inactivated, which means that this cyclic GMP is holding this Na channel open, which means that our voltage-gated calcium channel is being kept open, which means that we are secreting glutamate onto this bipolar cell, which means that this G protein coupled receptor is inhibiting this calcium channel, which is preventing the release of glutamate onto this ganglion cell here. So that's what happens in the dark. And that is how we turn light into action potentials. So now we have this phenomenon called receptor fields. And this is how these photoreceptors work together to produce contrast and sharp vision. So in a receptor field, say we have 3 photoreceptor cells here, 2 horizontal cells, and then we have our bipolar cell and our ganglion cell. So let's start with a couple rules here first. So dark conditions are always going to cause the release of more glutamate, and vice versa. Light conditions are going to release less glutamate onto the bipolar cell. And then our second rule is that our receptor fields are named by light's effects on them. So let's say for this example, we have an on center and then off surround receptor field here. So that means that light is going to turn this center receptor on, and these off receptor, and these surround receptors off. So in this case, when light hits this channel here, when light hits this photoreceptor cell, due to the events that we just talked about over here, we're gonna release glutamate, or we're not gonna release glutamate onto our bipolar cell. And then because of that, we're gonna release glutamate onto our ganglion cell, which is gonna send some action potentials to the brain. And then if we have darkness on these 2 surround cells here, what's gonna happen is that since dark releases glutamate, we're gonna release glutamate here onto these horizontal cells. And glutamate being released onto these horizontal cells is going to cause GABA to be sent to the center cell here. And GABA is going to further inhibit the release of glutamate onto that bipolar cell. So we are under these conditions, on center, off surround, we're releasing the most amount. We're releasing the least amount of glutamate onto this bipolar cell, which means that we're releasing the most amount of glutamate from this bipolar cell onto our ganglion. So let's say that in our receptor field, our center photoreceptor cell is responsible for 60% of the action potentials that reach the brain, and our surround cells are responsible for 20% of the action potentials that reach the brain. So if we're looking at something and say that we have an on center, off surround conformation, or a constitution of this receptor field, on center, off surround, and say we have this shadow here made by my hand on this left side of the field, and there's light on the other sides. So that would mean that we're going to create 80% of our possible total action potentials because we're on center, off surround. So since this side of the receptor field is dark, this is going to be contributing to our possible action potentials. Since the center is in the light, it is also going to contribute its 60% of its total possible action potentials. But since this side is in the light as well, we're not going to be contributing those 20% of our possible action potentials because these get turned off by the light. So that's the mechanism by which we perceive contrast and our sharp vision. And we can have bipolar cells that get turned on by glutamate from photoreceptor cells and bipolar cells that get turned off, so to speak. And so this is why we have our on center photoreceptor cells and our off-center photoreceptor cells. And due to these variations, that's going to give us even more ability to perceive contrast and sharp vision. All right, so that's my video essay there. So just let me check and make sure that I got everything. And I think we're good to go for this unit. So I described the chemoreception of smell and taste. I described how energy carried by sound waves turns into mechanical vibrations, fluid waves, and chemical signals, and action potentials for the things we hear. I explained how the eye focuses light. I mentioned the ciliary body and suspensory ligaments. I described the two types of photoreceptors, and I explained how they converted light into electrical energy. And then I explained how center and surrounds cooperate in a visual field. And yeah, I described the mechanism of hearing. I included the middle, inner ear and their roles. And I discussed how the hair cells produce action potentials. Okay, cool. Well, we are all done, so thanks for watching. Have a good one.